Everyone, welcome back to day 3 of the INCF 2021 DataJoint workshop. My name is Tin Nguyen, and I am a science project director here at DataJoint, and I'll be leading us through this day 3 of the workshop. So let me first start by sharing my screen. All right. Can you see my screen?
Yes.
Okay, great. All right. Sorry, I'm organizing my window so I can also see the chat at the same time. Just kind of move things around. All right. Yes. So yeah, again, welcome back to day 3. And so in today's workshop, we will be covering kind of using DataJoint elements, and we want to focus on specifically the workflow array ethos. And so as you've gone through day 1 and day 2, hopefully you would have a sense of what DataJoint is and some hands-on kind of interactive tutorial on using DataJoint. So okay, right. So the outline of today's talk, I'll be going through first a quick recap of day 1 and day 2, introducing DataJoint elements, a recap on Dimitri's talks yesterday on what DataJoint elements is, and also going into presenting one element, well, actually one workflow that is using these DataJoint elements, the workflow array ethos, focusing on extracellular electrophysiology type of experiments. And then I would go through quickly the outline of the workshop, and then we can just jump straight into the hands-on tutorial workshop for today. All right. A quick recap on day 1 and day 2. So in day 1 and day 2 for the lecture, hopefully everyone's have a good sense of what DataJoint, the DataJoint framework is, why is it useful, and what are our what DataJoint, like what are some of the other type of project and services that DataJoint offer, and what are our kind of like upcoming future directions for DataJoint. And all right, so let me start first with a quick recap. So what are data pipelines? So why are we doing all of this thing, right? So as you are aware, currently scientific research, especially in neuroscience, the data are getting just more, first of all, richer and much more massive, more multimodal type of data. One experiment, you would structure your experiment with multimodal type of data with a lot of different aspects of the experiment, and you want to piece everything together, running through different step of your analysis. So as data are getting richer and more complex, you would want to typically research lab and multi-lab collaborations are scaling up their data management, right? A better way, a more robust way and transparent way of managing the flow of the data through the project to the research. So really, what are data pipelines? I think we've been using these terms a lot, and everyone's means slightly different things. I think generally we agree on what we mean, but what is it? So, all right. So how do you typically describe your experiment? I'm speaking here, I guess, also from my personal view as well, but generally I would describe things as, so I start my experiment with some sort of a subject, so maybe an animal, and then I would perform some sort of an experimental session on it, where the animal or my subject would go through some sort of behavioral paradigm. So either watching something on the screen or responding to some auditory stimuli, things like that. And then from that experiment, I would perhaps collect data from a number of neurons. I would perform some sort of analysis on those neurons. I would have a set of parameters, and then going through some analysis routine, maybe calling different packages to do spike sortings. I would arrive at a spike train for my neurons, and then I can do further statistic on it, group level analysis, those kind of things. So all of these components, right, pieces in my experiments all together and also the steps in which I do things, right? All the sequence of steps where things are being carried out. All of these together make a data pipeline, right? So yeah, so in short, a data pipeline should first of all describe the things in your project. So animal, session, behavior, neuron, spike, statistics, some parameters. And also a data pipeline should also capture the relationships between these things in your project, right? Essentially representing a sequence of steps where how you would carry out your experiment, and also your experiment, your processing, your analysis, right? So how do you typically run your data pipeline then, right? I would imagine something like you have some sort of an acquisition system that would collect raw data, store data in files or directory folder with some hierarchical structure, with some sort of a directory or file naming conventions. And maybe those are for the raw data. And for the metadata, maybe you store that in maybe you handwritten it down in some data lab notebook or something, or maybe you have Excel sheet that you type in your meta information, subject number, session number, some sort of a note or set experiment type, things like that. Maybe there's text files, config files. And then you would have a collection of scripts, Python script, MATLAB script, or packages, tools, packages that you would run through run your analysis on. And maybe there's a sequence of them, right? So you would chain your analysis in different stages. And at the end of that, once you've run through your analysis, you would have some set of files representing your analysis results. And then you store that, and you can look at that, use that to interpret your scientific results and answer your scientific questions. And maybe you would then copy that set of files and share that with your collaborators, email it, put it on a hard drive, give it to them, something like that. Right. So as you can see, there's multiple steps, and a lot of these steps are captured really in the mind of yourself, like or myself. This is actually roughly how I did it in my study as well, right? So very multiple steps, all stored in my memory. Essentially, I know how this is run. Myself and maybe a few of my collaborators, my colleague. Okay, but then so the next question is, how do you scale this data pipeline? So a lot of these are kind of sort of manual steps, sort of undocumented steps, like the script that you run, the order of the script that you run, and then a lot of these are moving data, piping around, if you will, right? Moving data around. How do you scale your project to a multi-lab or share it with collaborator or co-working together with your collaborators from different institutions? So what I'm showing here is one kind of a data pipeline architecture from an example from a group that we've worked with. So as you can see, there's a lot of moving pieces here. First, the experiments are being done in maybe different labs or different institutions where the meta information can be captured via some sort of a web interface, like a web interface where you specify meta information, annotation. The raw data can be acquired locally, stored locally, and moved to a cloud-based infrastructure, right? So either a database server, like, as you're fully aware, DataJoints are built on top of a relational database. So at some point you have to move your data to either a database or kind of like a bulk object storage, like S3, for example, or Google Drive, things like that. And then a lot of the analysis can be done also on cloud, right? There's a compute server that would basically run through the set of analysis, the script that you specify essentially, and store the data back into the database server, for example. And all of that can be accessed by all the users from your collaborations, multi-institution, multi-labs, can all access that same data concurrently, make changes to the database concurrently if need to, define downstream analysis, visualize the results, all facilitated on cloud, if you will. And in this architecture, there's room for optimization, like parallel, like performance, right? So parallelizations are native, built-in. You can parallelize your analysis. Right. So this is roughly one example of how you can scale your data pipeline from everything in your laptop, right? On your laptop, you set up, files and raw data and script. You run everything on your laptop to how you can take it to the next level and do things in a more multi-lab or multi-institutions collaborative way. Right. So next, so what is DataJoint framework? As we've discussed in the past 2 days, I'm sure you're fully aware of what DataJoint is, but I just want to have a quick recap on on high level, what it is. So nowadays, I think in scientific computing or in research, there are two powerful mechanisms where you can design your pipeline, right? So you can do it using a relational database system or some high-level programming language, or a combination of both, right? So for example, Python or MATLAB, you can work with files, you can really prototype your analysis very quickly. Okay. In terms of the relational database system, these are very good for storing complex data and metadata. They offer structured data. Everything's are captured in tables with attributes and dependencies. They relational database system ensure data integrity and enforce it. So things a downstream table an entry in a downstream table have to exist only when there's a corresponding upstream table, right? So you have to have an animal before you have a session, something like that. As you've gone through yesterday or the day before workshop with David, I'm sure you've already seen that firsthand. Okay, and a relational database system support flexible queries. You can search things across your entire database. You don't have to do loop. You don't have to write loop through your files, open up every single file, search for things in there. Once in a relational database model, you can query things across the entire database, right? Across subjects, across sessions, things like that. And obviously, the most important aspect is that this is support concurrent access, both read, write, delete, things like that, right? So you will never be out of sync with your colleague, right? If you're looking at one set of data, your colleague from another institution, for example, would also be looking at the same set of data. All right. However, some drawback for these relational database systems is that they're not very friendly. They're not really meant for sophisticated computation. You cannot write new analysis script on them very easily, and they're not very friendly for neuroscientists. I'm not sure about you guys, but for myself, writing SQL statements is really, quite a pain. All right. On the other hand, we can, really brainstorm a little bit on how MATLAB or Python or these kind of high-level programming language would offer, right? So they're definitely good for sophisticated computation. You can write really sophisticated routines for machine learning, for complex type of analysis. They're very friendly to neuroscientists. Actually, I think we're all trained on using these type of high-level programming language. However, these are not meant for complex data storage. You can store things in Pandas data frame, for example, and then write that into an Excel sheet, things like that, or maybe write NumPy arrays or MATLAB .mat files. But really, essentially, at the end of the day, these are files. And because they are files, they don't really support queries either. All right, well, DataJoint framework is a combination of these two. DataJoint is a workflow management framework that offer data storage in relational data model, data and metadata, full specification of dependencies, with distributed computation built in. So hopefully, as you've already seen from yesterday, for DataJoint, the code to generate your data is embedded inside a pipeline. And because of that, DataJoint offer built-in distributed computations. Once you define, the MEC function, if you remember from yesterday, once you define that one time, you can distribute the computation to 5 machines or 10 machines or however many, or you can deploy the jobs to computer clusters, and they would all be DataJoint would manage how the jobs are doing distributed parallelizations, running your analysis much more efficient. All right, so a quick recap on DataJoint architecture. So really, at the end of the day, there are several aspects to it. First, there has to be a database server. That's where your data live. And then there's a data pipeline. So this component right here are your data pipeline, right? A set of tables, schemas and tables, and their dependencies specifying how you run your project, how you do your experiments. And then on each of the user computer, you can interact with this data pipeline via the DataJoint language via Python or MATLAB, and you can all share your work, query the same thing, insert data, and run further analysis on it. All right, so just a quick recap, right? DataJoint is an open source framework for scientific workflow management. These are supporting a framework for building and running data pipelines in Python or MATLAB based on relational data model, ensure and enforce data integrity with very intuitive and flexible queries. And the most important things is that DataJoint, being different from a simple database server, is that this is DataJoint is a workflow, right? So steps, the steps in which how your data flow through this pipeline, and also the code in which how each tables can generate entry for itself. All of them are embedded as part of the pipeline with built-in jobs distribution for parallelizations. All right, well, sounds good, right? Okay, great. So what's next? What are the challenges? Well, there's nothing perfect, right? There are a number of challenges in working with DataJoint for building your data pipeline. Well, not just DataJoint, but just overall any data pipeline, really. There has to be a component where you would have to go and design your pipeline, right? This design and implement your pipeline. This speaks to the fact that you have to specify, my experiment has these many tables, this is the steps, this is the code that would run this table, this is another code, another script that should run the second tables, and so on and so forth, right? Pipeline design, database designs are not a trivial task. And you then also have to define and write your processing and analysis script and attach to the table or orchestrate that in some way. And then there's this aspect of running the pipeline. So day to day, making sure that data are flown through your pipeline. Every aspect of the pipelines are being computed, operating and monitoring the pipeline. That's also not a trivial task either. And then there's also some work associated with database administration as well. At the end of the day, these are where your data lives is actually on a database server, so you have to do some sort of database administration. So things like user access, backup, things like that. All great things, all important things, but you have to do it. All right. So that brings us next to DataJoint elements. Actually, let me take a step back and kind of elaborate more on this. So as you can see, even for every single piece of this, there's actually quite a bit of work and I hope that as you guys went through day 1 and day 2, you see that how we have to write code to define your tables, define your analysis function for each table, things like that. So that takes work, and that takes good design as well. So we've worked with a lot of groups, and there is also many other groups that use DataJoint for defining their data pipeline. And these are all great starting points for a new user or maybe a new lab start setting up. However, these available pipelines out there, the way they work is a little bit too specific to that project or too specific to that lab, right? So you're welcome to take the code. They're all open source. You can go through it. But then I think soon you realize that, oh, a lot of these are just specific to this lab or just specific to this project. I don't need that. I only need like these 4 different things, but I cannot just copy the whole thing because it's just built in that way. So because of this challenge, recently, actually a year ago, DataJoint, we have been awarded a 5 years NIH grant to develop DataJoint elements. We term these DataJoint elements. The idea here is that we want to package these solutions that other labs and other institution have worked on. We learn from them, and then we package their pipeline into modular components where then we can turn around and offer it to the community as ready to go solutions where you can just take the whole, we call it elements, and I'll get into it a little bit more, and then you can use it for yourself. You don't have to read through 5 different code base and just extract the things that you want to use and re-implement things for yourself. These are ready to go solutions targeted for different neurophysiology modalities. All right, so what are DataJoint elements? And these are a set of predefined templated DataJoint schemas and tables fully describing a certain data modality. So things like your colony management, for example, right? Subject, genetic information, animal caging, things like that. Electrophysiology, right? Describing the probe, describing the EFIS recording, describing, the step or the tables that you would go through. Spike sorting, for example, waveform generation, things like that. And calcium imaging, defining how the steps that you would go through after you get your scan, segmentation, motion correction segmentation, or arriving at your fluorescent traces. What do you do next? Things like that. So we have a set of DataJoint elements, one for each of these popular data modalities. So these are independent and modular designed. These are essentially pip installable Python package that you would import, and then you can connect downstream. You connect these elements downstream from an existing pipeline. And along with each of these elements are built-in integration with other tools and packages. For example, and I'll get to the example later, but for example, if we are specifying the workflow array EFIS, we support a certain number of analysis package, right? Spike sorting packages or software acquisition packages. All right. Okay, so let's start with what I have here is an example of what one elements look like. So I'm showing here an example of what is the imaging element. So the imaging element is just this part right here. Downstream from it, as you can see, these dash line represent the connection point. So this imaging elements really connect downstream from these 3 tables. So I'm assuming that you have some sort of an upstream pipeline. You can then pip install this imaging element, import it, and connect it in downstream from your existing elements, existing pipeline. And this element offer these set of tables defining a typical calcium imaging experiment. So things like you have scans, scan info, processing, motion correction, segmentation, fluorescent classification, trace, things like that. What it is really still DataJoint. These are all DataJoint tables. For example, this is a scan table. This is how it looks like. And the usage is fairly straightforward. Once you have these imported, you can just activate these elements. So let me explain a little bit on what that means. These elements are just, like I said, templated set of tables, meaning they're just really code. They're not yet tables. They haven't yet connected to any real database server. There's not a tables on any database, on any MySQL server anywhere yet. These are just templated code. Once you import them, you can then activate your elements by running something like this, right? Imaging.activate. So here, like what I have here is an example. Like once you have a pip installed, you can do like from elements imaging, import, scan, and imaging. Right? These are the 2 modules in this element. Once you have that, you can just do something like, imaging.activate, and then specifying the database connection and things like that. That's essentially instantiate all of these tables on your database server. Now you then have access to all of these tables that have already been defined, and then you can just work with them as if you've already gone through the work of defining all of these tables yourself. And mind you that for each of these steps, we also introduce, code that goes with each of these tables. So these are imported tables. These are computed tables as David gone through with you guys yesterday. we also provide like for each of these elements, the code that goes with each of these tables that knows how to populate itself. So essentially all you need to do is specify some manual information upstream and then just run the populate routines to get your table populated. And we'll go through all of that later in this workshop. All right. Okay, well, the next one is another example of another element. This is the EFIS element, or the array, the EFIS array, and the element array EFIS. And as you see, it's just this component right here, this part right here. And there's also an upstream component where the EFIS element is being connected to, right? So as you can see, all these are all, again, all DataJoint schemas and tables defined as fully defined a extracellular electrophysiology type of experiment. And same thing, there's these DataJoint tables and the way you would activate these elements, right? So, all right, okay. And so what I've shown there are basically two elements, and I just want to quickly show an example so that you have a sense of what these elements are and how to use them on a high level. Moving forward, so DataJoints are going through a 5 years U24 project where we would, our goal is to define and present and package and present a lot more number of elements. There will be one for animal management, instrumentation management, trial-based type of experiments, right? Behavior, optogenetic perturbations, extracellular and intracellular electrophysiology, functional imaging, calcium imaging, machine learning modeling. So the goal for the 5 years goal for us is to present and prepare and present these elements to the community where they can just build your workflow, build your data pipeline from these elements. So keep in mind that each of these are standalone, independent Python package, right? You can mix and match, right? To put together a workflow, you can essentially just pick this element, so the animal management element, the lab element, and the functional calcium imaging elements, and just those three, you can put those three together into one workflow that works for your labs. All right, so the focus of today will be on the workflow array EFIS. This is a workflow that has been put together using 4 elements, right? So I think it's the, yeah, we'll go into each of those elements and how they're put together and how they work. But for this presentation here, I just want to quickly go through with you on the high level. What are these and why we are doing it? Okay. So what I have here is a simple diagram on high level. What's a typical extracellular electrophysiology type of experiment? So in these type of experiments, you start with an animal. Oh, actually, sorry, I see that there's a question or chat. Oh, okay. Hi, John. Yes, thanks for the questions. The elements and workflow are written in Python for now. So far, we've put a lot more focus on the Python side of things, and yes, they are written in Python and they are operated in Python. But being DataJoint tables, you can just work with these elements in MATLAB as well, right? So once you have your pipeline defined, so to start out, you have to do some work in Python. But once all of those are set up, you can access these tables in MATLAB, the same ones, right? You can access these tables in MATLAB. You can extend these workflow. You can add your own tables in MATLAB. You can work with the rest of it using MATLAB perfectly fine. Okay. All right. Oh, right. Going back. Yes. Right. So these type of experiment, typically you start with an animal, right? Your subject of interest, and then you would perform some sort of surgery. You would place an electro array or multiple of such electro arrays, or here we call them probes, into the certain brain area of interest, and then you would start collecting neuroactivity from these probes. In our workflow array EFIS, we support NeurPixel probes. So NeurPixel probes has been kind of like the focus of review in the past several years. They are kind of like a great engineering effort where now you can actually essentially record from hundreds of channels on a single probe. NeoPixel 1.0 offer up to 800 something electrode sites. Actually 960, yeah, actually 960 electrode sites on the probe on that one single shank. From those 960 sites, you can select up to 384 channels to record from. So across that shank, you can say, I want to get like 100 channels from the top part, and then another 100 in the middle part, and so on and so forth. So with such a high, a large number of electrodes, you actually get a great detail level of of neural activity recorded from any brain region of interest. And then NeoPixel 2.0 recently released, or soon to be released, I think, offer up to 4 shanks, and in each shank you have north of 1,000 electrode sites, in which you can all select from those 384. So the brain coverage is much higher. The number of potential neurons that you're being able to record from is also great. So so in this experiment, essentially you would then perform some sort of surgery to place these NeoPixel probes inside one or a different number of region of the brain of interest. Maybe you can place 2 or 3 or 4. I know that some labs are doing 5 probes at a time. And then from each probe, you would then acquire essentially EFIS recording from each of the probe, right? As the animals are doing some sort of behavioral task or something, you start acquiring electrical activity from the brain. And the workflow array EFIS that we have support 2 types of acquisition software: SpikeGLX and OpenEFIS. These are actually the 2 leading type of acquisition software for NeoPixel probes anyway. So if you are using NeoPixel probes, chances are you are using one of these 2. Right, and then once you've collected your EFIS recording, the next steps are typically performing some sort of spike sorting. So maybe I can take a quick step back and pause a little bit here and explain like usually what that means, right? So what you get from the raw recordings are electrical activity from each of these 300 channels, from all of the hundreds of thousands of neurons in the surrounding tissues, brain tissues, where your probe is. What you're interested in, or what neuroscientists are typically interested in, are the actual neural activity from each of these cells, right? From each of these neurons. But what you collected are not really activity from the individual neurons, but more like a superimposed collections of all the electrical activity from all of the surrounding cells. So a typical step for this type of experiment is to run through some sort of an analysis that would help you to extract neurons and their individual activity from this mass, superimposed set of data sets that you collected. Essentially something related to something similar to the blind source separation type of problem, right? And there's a number of different packages that do this kind of spike sorting. One that we support in this workflow is KiloSort. So KiloSort is a quite popular package for spike sorting, and they've done a number of optimization for the NeoPixel probes. Okay, so once you've gone through your spike sorting, or usually referred to also as clustering, spike sorting clustering, I'll be using these terms interchangeably. So once you've gone through the step of clustering and then arrive at a number of cells and their electrical activity, you may or may not want to do some sort of manual curation. So this is what this step represents. So manual curations refer to the step that you look at your results, and then you decide, okay, these are not very good. I'm gonna manually change something. I'm gonna exclude this cell because it looks like noise. I'm gonna merge these 2 cells because they probably are the same. Things like that, right? So manual step of checking off the list, cleaning your data. Once that's done, really at the end of the day, you arrived at these kind of data, right? So you have a set of units a set of neurons, what you think are what you would think are neurons. So that's why we're usually in the field, it is actually called unit and not neuron, because you're not really sure if that's a neuron or not. Yeah, so a set of units, and for each a spiking activity for each unit, and also the associated waveform for each unit. So from these, you can do a lot more further analysis on this, right? You can segment these, you can align with some interesting behavior that you're running, you can cluster them, all kind of downstream analysis, further analysis to answer your specific scientific questions. But a lot of the steps really are ended at, perform the surgery, put the probes in, do some data collections, and then do some spike sorting, arrive at your cells, your neurons, and then do further analysis on that. All right. So with this, we've worked with many different labs that running these type of experiments, and for each lab we help the labs, and they each have their own DataJoint pipeline for this very same set of experiments, and each has slightly different variation of the DataJoint pipeline. So as a part of the U24 effort, we put together a DataJoint workflow to model this type of experiment. Okay, so for the rest of today, we'll be going through this workflow array EFIS. This is built from DataJoint elements. These are the 4 elements that we're using in this workflow. So elements lab describing the lab, animal describe the subject, right? The mouse, session, and elements array EFIS. This is the bulk of the pipeline defining the steps that I've just gone through, right? This diagram right here. Okay, this workflow array EFIS support comes with ingestion routine that support NeoPixel probes 1.0 and 2.0, Spike GLX and OpenEFIS acquisition software, and KiloSort as the spike sorting package. And come with this workflow is also the capability to export data into NWB format. We're still actively developing the best way to export into NWB format, but the ultimate goal is that for every element should know, every DataJoint element should know how to export data into NWB format. And if you're not familiar with NWB format, it is neural data without border format. It's essentially an effort of defining a format for storing neurophysiology type of data. I think neural data without format is also holding workshop for this INCF 2021. I would definitely encourage you guys to check that out. All right. Okay, so this is it. What you're looking at here is the entire workflow array EFIS, DataJoint workflow array EFIS. So as you can see in the middle, I'm showing DataJoint table, right? So the different variety of DataJoint tables to model this entire process. But really, what the takeaway here is that each portion of this set of pipeline relate to a step that I was describing a few slides back, right? So there's some component describing the probe, the NeoPixel probe, what they are, what are the channels, the location of the channels, and things like that. There's component of this pipeline that describes the probe insertion, the act of surgically place the probe inside the particular brain region to do recording from. A setup table describing the EFIS recording itself, right? So collecting data at, 30,000 hertz, storing data somewhere, and things like that. And then there's these steps that describe the spike sorting and the manual curation that comes with it. And the setup tables describing the final result, the unit spikes waveform, the final result that everyone would want to look at. So this pipeline right here describes essentially the experiment and the data management aspect of your experiment. It stops at the final product where you would then do further analysis on, right? You would then design different type of analysis to answer your scientific questions. And a lot of different labs are redesigning this DataJoint pipeline for their use in the past few years, and it's kind of like at some point it becomes an inefficiency and part of the U24 effort is for us to consolidate those knowledge and put together a standalone pipeline for everyone to just use right away. All right. Oh, click the wrong button. Okay. In my next slide, I am essentially zooming into this pipeline, and I want to step through it on a few key tables that we want to touch that should be of importance to you, right? First, we have a set of table describing the probe, right? So there's probe type. These are like NeuralPixel 1.0, the NeuralPixel 2.0 single shank or multi-shank. There's a probe table. This describes a physical probe that you actually use for your experiment. You know, things that, you know, I've purchased this NeuralPixel probe. This is the serial number. Let me block it here so that I know that I have that probe. And then you have a notion of subject, session, probe insertions, where you perform the surgery and put the probe in. And if it's recording, you run the actual recording, acquire raw data, where those files at, local field potential. This can be extracted, or this can be part of the raw data that you collected. A number of tables defining the clustering tasks and clustering. This is the spike sorting step. And as you notice, above this, we have some lookup table defining the method that we want to use for this clustering, and also the parameter set that we want to use for this clustering. So this can be something like kilosort 2.0 as the method, and some big dictionary for all of the parameters that you want to use, that you actually use to run your spike sorting. Then there's a step of curations, and then some imported table defining loading the data in. So these are the units, the spikes, and the waveform, things like that. All right, so let's talk about using the workflow, and we'll later on we'll actually do some hands-on workshop to actually use it. But right now I want to give a high level description of what would you typically do as an experimenter for your data, right? So this is essentially the pipeline, like a truncated version of the pipeline that you would probably be interacting with the most, right? As you can see, these are mixed up DataJoint manual table and imported tables. So these are manual tables. The green ones are manual tables, and the kind of blue purple ones are imported tables. And if you remember from day 1 and day 2 workshop, manual tables and imported tables have the distinction of, well, as the name implies, right? Manual tables are meant to store the type of meta information where you would manually insert them. Either manual in the sense that either you have like a Excel sheet somewhere that you would then import and insert in, or you have an online web interface where you would type, you know, session number 4, subject name Bob, or something like that, right? So those are the type of manual table. Imported tables are table designed to be importing information from acquisition data, so file. So you have raw data and some collection of files. Those are then imported into these imported tables. So as you can see that there's a mix of manual and imported tables. As you would have guessed, running this pipeline involved a combination of running the dot insert and dot populate, right? So the manual table you would insert manually using the dot insert as we've gone through in day 1 and day 2. Getting in data ingested into these imported tables involve running dot populate, right? So when you define your make function, you just run dot populate on those tables. Okay, so essentially, the step would be something like this. So first, you've got like a new animal. You can do like dot insert your new animal in here. Oops, clicking it. Then, sorry, give me 1 second. Let me try to get back into presenter mode here. I think I am. Okay, great. Next, Then you would perform some experimental sessions. And then you can say, okay, well, I've just finished running a new session. The acquired data can be found here in this directory. You insert that information into this table. And then in the probe insertion table, you say that, well, for that session, I actually used 2 probes. These are the serial numbers for the 2 probes that I used. And these are the brain region also, for example, that the probes were inserted at. Then you would run some auto-populate routine, right? So DataJoint table would know how to like go to those data set and load data. Same for the local field potential data. Then you would perform some sort of splice sorting, right? You run through your data using kilosort. You've arrived at here you can specify, okay, I've just finished running through my splice sorting using this method and this param. The data, the sorted data can be found here in this directory you specified here. Next, you can run, again, right, the auto-populate for this table, being an imported table, this table can go and check this folder that you just specified, load a number of files and whatnot. Okay, and then you may or may not do some curations, manual curation to your data. You can then specify, okay, well, I've done some more manual curations. The curated data is here in this directory. Go look for it here. Then you can run .populate here and here, and the rest of the pipeline would know how to import the data, load the data into your tables, ready for you to query the data and work with the data. All right. Okay, so that's pretty much on high level the overall design of the entire workflow array EFIS and the step that you would actually take to run through your that pipeline. So hopefully, as you can see, that's really kind of mimicking how you would run your experiment anyway, right? There has to be a step of defining, oh, this is a new session, defining I'm doing recording from these 2 probes in the brain. These are the data that's been collected. Now, next, I'm going to be running analysis, splice sorting. This is the result. This is where it is. Some curation step. These are the result of the curation step. Here it is. And then go load my data. Essentially something like that. All right. So the interactive workshop that we'll be doing next will be actually a hands-on tutorial on how to do this for your data. So with this, the goal is for us to go through and actually use workflow array EFIS to manage your EFIS experiments and learning how to work with your data from the pipeline. The resources in terms of resources, we have prepared a Jupyter server ready for use, and you can find it here. You can click on the link here, and this Jupyter server already have the workflow array EFIS pipeline installed. So you don't have to go through all the steps of, pip install different packages, put the structure together and activate them. These are already taken care for you, right? And then along with the part of the resources is actually a database server, the same one that you've been using for day 1 and day 2, a database server. If you have DataJoint IO accounts, you can access the database server. This is the tutorial database. You can create your own personal schemas on it. And we also prepare some example EFIS data for us to play with. Right? Okay, so all of these resources here are provided to you so that we can easily go through this interactive workshop without you having to learn how to, bring up a database server, pip install different packages, having a well-configured Python environment to run this. All right. However, if you want to learn how to do all of this stuff from scratch, that's actually going to be our topic for tomorrow, for the workshop tomorrow, where Kelby would take you through. If you want to start from scratch, if you don't have access to any of these resources, what would you do? Okay. All right. So in the next 10 minutes or so, we'll start with the workshop. The goal of the workshop is to ingest data from subject 6 and session 1. What does that mean? It means that for the purpose of this workshop, we have already uploaded a number of sample EFIS data set from multiple subject, multiple sessions. Like this is essentially, you can put yourself in the shoes of an experimenter that have already gone through some data collection from 5 or 6 subjects with a number of sessions there, and now you're just going through the motion of getting your data into the pipeline. Right? How would you get the data into the pipeline so that it can be automated, populated, running further analysis on it? Okay, so we'll be using subject 6 and session 1 as our example. The date time for the session is this one, so 2021 01/15. I actually just made this date up, so really don't pay too much attention here. You can just change it to any other date, really. But let's just go with this one. So the idea is that you need to know your subject to make a new session. For the new session, you need to know the subject and then the session date. Then you also need to know where the session data is, right? So after you've collected your raw data, where is it? You have to point the pipeline to where to get that. So for this exercise, the session data is already uploaded, and this is the location of the data. But I mean, you can easily imagine that you may store your data in like C drive, my data, and whatnot. So something like that. Okay. In this example, there's only one probe that was used. And it's probe 0. The type of probe is NeoPixel 1.03b, and the serial number of the probe is this number. And that's a typo here. It's actually also probe 0. The spike sorting result for probe 0 can be found here as well. So these are the type of information that you as an experimenter and the data, like a scientist, you already know this information as you collected your data. So here, because we're using example data set that we provide you, we would also provide you with this information as if you're the experimenter, you already know this information. All right. Okay, so this is the end of my presentation here. Let me know if anyone have any question. I'll be happy to take any questions now. If not, and then we can take like maybe a 5 minutes break or something, 10 minutes break also, and then we can get back with the interactive workshop. All right. Any questions so far? Yeah, great. Okay, so yeah, these are pretty much high level. Once we get to the workshop, I'm sure we would have, like, we'll get our hands dirty, and then we'll probably have more questions here. Okay, so I will share this information in the chat so that everyone can refer to this, because this is essentially kind of the information that you would need to get your data ingested in the pipeline. Okay, but anyway, in the meantime, please try to access this Jupyter server and see if you have any, if you can access, if you have account. If you don't yet have DataJoint IO accounts, you can create accounts here. And maybe I can get out of this. Just for everyone to have a reference, once you access that link, this is the page that you should see. And then for our workshop today, it will be this one. It will be this server that you can click on and click start. And so I'll leave this here. Maybe let's take like a 5 minutes break and we'll come back to this. We can get started there. All right, yeah, so maybe a quick break. All right, so I'm back. Maybe we can wait 1 more minute for everyone to get back. If you're back and if you're ready, would you mind maybe put a thumbs up in the chat? All right, let's get started. Okay, yeah. Hi everyone. Welcome back to this day 3 DataJoint workshop. So in this workshop, we'll be focusing on learning how to you work with a workflow array ethos, a DataJoint pipeline built using DataJoint elements. And for this exercise, I actually, before I start, I can share the information that was specifying here on the chat so that you can always go back to it if you need to. Okay. Chat. Okay. All right. So for those that are following and try to we plan this to be an interactive workshop. So if you have any questions or anything, please just pause me and put your questions in the chat, and I'll get to it. Or yeah. Okay, so let's get started. So this is the server that you want to start. So you can, once you're here, you can click start. Okay, now you should be here. And what we have here is essentially a Jupyter server that already set up for you with the workflow array if it's already installed. So you can think of this as, you can put yourself in the shoes of the experimenter or a data scientist that already have all of the environment for development already configured for you. You have already a database server, maybe your IT team set it up for you, or you set it up yourself. Essentially everything is ready to go for you. Now you just like get to work, right? That's kind of the mentality that maybe you can pretend yourself to be that person. Okay, to start with, you can click on this workflow array ethos right here, and in here you would see basically the entire repository for this workflow array ethos. But we prepare a number of notebooks for everyone to follow to step through to learn how to work with this pipeline. For 2 days, we'll be focusing on notebook 1. Just configure your credentials, your connections, and things like that. And then notebook 2, a quick walkthrough of the actual structure of the workflow. Very much the same stuff that we have already covered in the slides that I presented, but let's just do it again, let's step through it quickly again. And then notebook 3, this is going to be the primary, one of the primary focus of the workshop. Notebook 3 showed you how to, assuming that you already have your data collected, how do you get your data to this pipeline? And then how do you get all of the ingestion routine running, all of the automated processing going? And then finally, we're also going to be going through notebook 5 as well. What do you do after you have your data in your pipeline? So notebook 5 will be going through exploring your data, working with your data, retrieve your data, and do some quick plots on your data. Right? Just as a demonstration of how what would you do once all the data are there. Okay, so let's get started. So here I've launched notebook 1 and a new tab. So you can start stepping through this with me. So the first step is here I'm doing a change of direction of directory going 2 level out, but really you don't need to do this because a lot of these steps are designed so that if you're doing it from scratch by yourself, you would have to do some of these steps just to get your configuration set up. But here I just need to import DataJoint, and you can just follow along with me as well. So first I import DataJoint, and then I would specify this next cell here is specifying the host, the database host, the username, and the password. But before we do that, I want to show everyone quickly, because this is a Jupyter server that we have already set up for you guys. If you do dj.config, you will see that the database hosts have already been predefined for you. So this is the tutorial database that we've been using over the past few days. So you can just copy that and use that for your host. Now, if you were to do this by yourself locally, then obviously you need to change this to whatever is the database server that you have set up. Either that being the local host if you set it up yourself locally, or maybe a different address that your IT team set up a dedicated database for you. Right, so username, so it's my username, and if I run this cell, so once you specify those, you can run this cell, right? If I run this cell, this would ask for my password. So I'll type in my password here. Okay, enter. And then I can try to connect, see if I can connect. Okay, awesome. Yeah, so I'm in, so I'm connected. Oh, wait, no, I'm not connected. Wrong password. Okay, yeah, wrong password. Okay, now I'm connected. So if you see this, you're connected. All right, so the next step is that because we will be working with a new set of schemas and tables, one thing that you can do is to define a prefix for where your tables and schemas would be. So in this tutorial database that we set up for you, everyone should have free access to any schemas that start with your username. So for example, what I'm going to do here is I'm going to put my username and I can put underscore. So anything that start with this, I would have access to. So for this example, let me do this. Yeah, I'll be using this as my prefix. So essentially what that means is that every single schemas and table that I will be creating for this workshop, for this workflow, will be starting with this string right here. So you can do the same, right? You can put your username here, and then you can do underscore ethos underscore if you want to. Okay. Can you run this line? All right. So, and then the next step for us is we will need to specify where is the root data directory that we are storing our data at, right? In this example, we have already uploaded all of some example data set in a different location in, and this is the location here. So it's under temp, test data, and then workflow array ethos dash test set. Okay, so this is where we have already uploaded a number of data sets for everyone to try here. So you can specify this here. If there are multiple roots, then you can use this notation right here, but really we don't need that. We would just be using this one. So I'll be commenting that line now and just run this line, right? So this is what it does is that it would store this path onto your config file so that on your config file so that you can have access to it anytime later, right? And it's basically tell the pipeline that my data or all the raw data or really all of my data are stored here. At least the root of that are stored here. So if you wanted to look for my data, go ahead and look for it. Okay. let me run this as well. And I'm not going to run DJ config here because that would then show my username and password and everything. So let's not do that. But you're welcome to run this line so that you can see how the config is being done. What I would do next, now you can save all of this configuration. So you can do save local. That would create or save this configuration into a local file called DJ local config. Or you can do save global. So the difference between save local and save global is that this would create a file in your current directory. Save global would create that same config file in the root directory of your user for this whole system, so you can reuse it any other time. So for myself, I'll do a save global, and yeah, for this exercise, please also do the save global. Okay. So all of the configuration that I've just specified have already been saved globally. All right. Okay. So let me reconfirm and just print this out. See if, yeah, I can just do it here. Okay, so temp test data workflow, blah, blah, blah. Okay, so let me clarify again. This is the root directory of where all of your data essentially are stored, right? So this can be a directory where your acquisition system would just split the data there, and then you can just point, you can just set this configuration to that file, or you can imagine this being a mounted drive, right? So you have, if your institution or your lab has a network drive that store all the raw data, you can then specify this to be that mounted drive right here. So let me just quickly show everyone what's really there, right? So let me just CD myself here. I will, okay, so here, even in Jupyter Notebook, you can do simple like command like change direction, change directory, Linux command, and then you can do like ls. Okay, so as you can see, now that I've navigated myself here, if I do like a list of everything, I see that the subject 1, 3, 4, 5, 6, and so on, and some other files. Because for this exercise today, we'll be focusing on subject 6. Let me just CD myself, change directory into the subject 6 directory. Okay, I'm there. Let's do another list. Okay. So as you can see, inside subject 6, there's a single folder called session 1. If I change directory into session 1, as you can see, there's another folder called IMEC0. This means this essentially is a folder for probe 0. So in this session 1, there's only a single probe that's being used, and all the data for that probe is in this folder right here. So as you can imagine, right, if you have multiple probes, then you probably would have multiple folders. So maybe that would be called IMEC0 or IMEC1, IMEC2, or probe 0, probe 1, probe 2, whatever. It's just some folder name for your probe. And if I keep on going, CD myself into IMEC0 and do list again, you can see these are all the files, all the raw data, and analysis results for this data set. So things like, you can see a number of files related to the kilo source routine, a number of files related to the raw acquisition. So these .ap.meta, things like that. Those are related to the raw acquisition. Okay. All right. Yeah. So I just want to quickly show you guys what is the actual directory where we have already uploaded some sample data set for you guys to use. But obviously, if you do this on your local system, then you would know where you have your data, what subject, what sessions. These doesn't have to be following us any certain naming convention at all. You can name this Bob. You can name, session 26 or CD at test dash data, not test underscore, John. Yeah, let me go here real quick. Yeah, so temp and then test dash data, and then yeah, workflow dash array dash efis test set. Can you try again and let us know if that works? Yep, awesome, right, yep. So yeah, let me highlight again on, there's no naming convention that you have to strictly follow here. Meaning if you want to try this on your actual data, you don't have to like know a certain like hidden naming convention or anything, right? This subject 6 can be Bob, subject 5 can be Tim or whatever, and then session 1 can be a date or something. So because at some point later we will specify where the data are at. So the naming convention can be whatever you want. In this case, we name these like subject 6 session 1 and so on and so forth, just to make things easier. All right, so this is the end of notebook 1. All that we do here is we just configure we configure several things, right? First, the credentials, database credentials. Secondly, this root data directory. This is extremely important because this is going to be where the pipeline would look for your data. So you can just do that, right? So that means that the root of my system is the root of my data.